input. I think, yeah, this is great.
So I think we will go ahead and get started. I just wanted to, again, say welcome and thank you all for coming today. We have our fabulous speaker, Dr. Braden Tierney, presenting about the Extremophile campaign in your home project, which many of you are already a member of. And if you aren't yet a member, then we encourage you to join and we can help you out with that. Just real quick, I think a lot of folks on this call are probably, oopsies, familiar with how these webinars work, but we do have a chat feature here and a Q&A feature. I'm not entirely sure which one's going to work best for folks, but I will watch both of those while Braden is speaking. And if you have questions or comments or thoughts, we want to hear them. So please use those features. And if something isn't working, I will be monitoring my email, so you can send me an email as well if you'd like. And with that, I'm going to let Braden introduce himself in just a moment, but I also wanted to call out James Henriksen, who's also on the call and is another one of the scientists on the team who will be here to answer questions as well. So we've got a great panel of folks, and I think you're going to all have a great time. So with that, I will hand it over to you, Braden. Oh, and I think I mentioned, but for those who are new, we are recording this so that other folks can watch it at a later time. So just be aware of that as well.
Great. Right. Let me queue up these slides, then we'll get going. Okay, let's see. There we go. Can everybody see that? Can I get a thumbs up or something like that? Good. Okay, awesome. Okay, hello everybody. Thank you so much for coming out in the middle of the day to learn about microbes and extremophiles. I'm really excited to share a little bit with you about the Two Frontiers project, about this project, and about some of the work we can all feasibly do together. So first, my name is Braden Tierney. I'm one of the co-founders and the executive director of the Two Frontiers project, which you're going to learn a lot about today. I'm a microbiologist by training, a scientist, and a data scientist. So I spend a lot of time thinking about bacteria and then all the ways we can analyze bacterial genomes, mostly like the genes they have with computers and large statistical data analyses and big things like that. And we're also joined here. We have James Henriksen on the call. James, I don't know if you want to introduce yourself very quickly.
Yeah, I'm an environmental microbiologist and a little bit of a bioinformaticist, using computers to analyze genomic data. And I really love the vast diversity of microbes and what they can do. And so I'm really excited to talk about some of those things.
Excellent. All right, so here's what we're going to do. Basically, it's going to take about 30 minutes, I think, 35 minutes of talking. Please feel free to jump in with questions or comments or whatever you like along the way. This is really meant to be an informal discussion about these three topic areas. The first one is microbes and why they matter. The second is the Two Frontiers project, what we're doing, why we're doing it. And the third is this project that brought us all together here today, exploring the extreme microbiology of your home. So first, I want to establish we do not live in a human world. We live in a microbial world. These microscopic, single-cellular organisms that you cannot see individually with your naked eye are everywhere. They're literally everywhere. And microbes have a bad rap, especially bacteria, because we usually think of infections or pathogens or negative things. But in fact, most bacteria are harmless, if not helpful. And so I want to talk mostly today about the ways they are helpful and where they are and what they're doing. So here we are. That's Earth. You're somewhere on this planet. We all are, right? And this world of ours, well, when you think about it from an evolutionary standpoint, so from the tree of life, so this is a diagram indicating how all organisms in the world on this planet here are related to each other. Eukaryotes, humans, all the life that you can see with your eye is way down there in that corner. The rest of this tree, everything else, these are all microbes. These are all microscopic life forms that we have discovered living on this planet with us, or we are living with them. And you can see on the left-hand side of the screen here, this is a plate used to grow bacteria where they've basically grown microbes on the culture plate in the shape of the planet. And it's kind of funny because, yeah, while you can grow bacteria, some bacteria in the lab like this, you certainly can't grow all of them because, the Earth has about 10 to the 30th microbial cells on it. Most of them we have no idea what they are. We have no idea what kind of species they are, what kind of genes they have, and we don't know how to grow them like you're doing on that plate. So 10 to the 30th, to put it in sort of easier to understand terms, is about 5 quintillion quintillions. So huge number, incomprehensibly large number. There's 1 billion microbes on Earth for every star in the observable universe. Just a milliliter of soil has, what is that, 100 million microbial cells and tens of thousands of species just in that little bit of soil. And so microbes make up 17% of the biomass, like on the planet. 93 gigatons of carbon. If you count living cells, the biomass of microbes make up 40% of the biomass on the planet. So the point is, this is their world, not ours. They're everywhere. If you were these are some of my favorite statistics to share with people when you're introducing microbiology. If you were the size of a microbe, about 1 micron, which is about 182,000-fold reduction in size from a 6-foot tall person like me, 1 millimeter is equivalent to a mile. A square meter of soil relative to that microbe is how the entire continent of Europe would feel to you. The arm span would reach from the glaciers of the US mountains to the coral reefs of the Gulfs of Mexico, and you could fit, relatively speaking, the entire surface of the Earth in your backyard, about 40 by 40 feet. So what does this actually mean? Well, it means that, if you're a single little tiny microbe living in a 40 by 40 area, like, think about the amount of variation in, I don't know, sediment and chemistry and temperature that can happen in, I don't know, your backyard, right? You could have sand in part of it. You could have clay in part of it. You could have grass in another part of it. That is like different mountains and seas and continents and universes to different microbes. And so in a very small area, you can have an incredible diversity of life. And these organisms, the billions and billions of them they're all very complex. Humans have about 20,000 or so genes in their genome, in our genome, right? That's what defines us, makes us human. It's what drives our life. Microbial life has billions of genes. There are literally billions of different genes with different functions that allow them to survive in these diverse environments. It's what makes them nature's alchemists. So it's funny, this is one of those diagrams that you'll see in science a lot that scientists love to flash up on a slide because they're like, look at how complicated it is. And no scientist, no one understands what this is communicating. It's completely uninterpretable. The only thing it's meant to show is like, these are all the little nodes represent different genes and functional pathways and things microbes can do. And so what this communicates, it's like an emoji. It's a scientific emoji. It's basically saying microbes can do lots of things. They can carry out all sorts of physiologies. They can capture carbon. That's somewhere in this corner of this diagram. They can breathe iron. That's somewhere in this corner of this diagram. They have this vast biochemical space. And so what this basically means, because they have this vast diversity, they have this vast functional diversity, where there is a molecule, a microbe will have evolved to eat it. So if there's energy being made in a chemical reaction, microbes will use it. This is what it means by like a microbe can breathe iron the way we breathe oxygen, or manganese, or sulfur, or nitrogen, or even currents. Microbes are responsible for producing half the nitrogen in the biosphere fixed from in the air, half the oxygen in the air from molecules of water. More than half of the CO2 fixed from the atmosphere comes from microbes. Now, really like think about that third point, right? Because this actually ties into what we're here to talk about today. So of the carbon dioxide in the atmosphere, half of it is fixed by microbial life. I have a statistic on a future slide that tells you how, 20 times the amount of carbon dioxide that is produced by humans, 20 times the amount of CO2 we make every year from your cars and everything is consumed by microbes in the ocean. So that means there are naturally evolved microbial mechanisms out there that are really, really good at cleaning up the environment. They already work harder than they already do so much more work than necessary, basically, to clean up what we produce. And so if we could just use that and figure out how those microbes are fixing CO2 from the atmosphere, we could feasibly clean up the tiny, tiny little bit that we add compared to all the other life on Earth. But these microbes aren't working alone. Microbial life is a team sport. They live in what we call microbiomes. This is an important word to know for the context of this webinar. Microbes, these are communities. These are like neighborhoods. microbes that live together and work together to survive in a particular environment together. So they have, for example, in your body, you have your gut microbiome. You might know this one. So this is associated with all these different diseases. The gut, the microbes that live in your colon, on your skin, and in your mouth can predict disease. They can actually be associated in some cases with causing heart disease or inflammatory bowel diseases, or protecting against autoimmune disease or causing autoimmune disease. They obviously affect gastrointestinal issues and bloating and constipation. Some of you might take probiotics, right? Those are used to change the microbes in your gut and help move your gut towards a beneficial microbiome. Microbes are also these microbial communities. These microbes are critical for coral reef health. We at Two Frontiers do a lot of work studying coral reefs. We basically are interested in how the microbes that naturally live on corals can actually make corals more tolerant to increasing global warming waters. So warming waters due to global warming. So effectively, the hotter the ocean gets, corals die, but there are bacteria that can prevent corals from dying in hot waters because they live on the coral. They are part of the microbiome. They are part of the organism, just like the microbes in your gut. This is that statistic I was talking about earlier. So microorganisms in the ocean fix 20 times the carbon dioxide produced by humans each year. This is another example of a scientific emoji. Like if a scientist ever throws something like this up in front of you, it's not trying to confuse you, or they're just trying to convey complexity. There's so much complexity out there. These microbes are doing all these different things to capture and produce and fix carbon into different forms. And so there are a lot of opportunities as scientists who study carbon capture to use these naturally evolved mechanisms to address sources of global warming. And so this, if anything, if you take something away from this, I hope it's a little bit of optimism, right? It's that we have these small microscopic neighbors on the planet. We share the planet with these 5 quintillion microbes that are doing really incredible work already at cleaning up the atmosphere, at maintaining what we call homeostasis. And so the mechanisms to fight climate change already exist on the planet. We just need to go find them. So that's section 1. Section 2, we're going to talk about the Two Frontiers project. So about going and finding those mechanisms. So that's us. So this is, you probably know the movie Jurassic Park. I love Jurassic Park. I love Jeff Goldblum. One of the famous lines from this is, "Life finds a way." At the Two Frontiers Project, we are interested in this question of how life is finding a way, especially when you don't expect it to. So the science of survival. So if you have a microbe that is surviving in, for example, a really high carbon dioxide environment, whether it be one in your house or in a volcano under the water, we want to know how it's surviving there. Because as we said, microbes are nature's alchemists. If there is a molecule, if there is a compound, if there is carbon dioxide, they will have figured out how to eat that carbon dioxide to survive. And so the entire premise of Two Frontiers is, let's go to these places. Let's explore these parts of the world where these microbes live that have evolved over billions of years to consume carbon dioxide, to consume methane, to consume other greenhouse gases or other pollutants. And let's figure out how we can use those organisms to actually solve problems caused by and faced by humans on Earth. So exploration and application of microbial life's evolution from the depths of the ocean to the reaches of space, humanity's 2 greatest frontiers. This is who we are, right? We work with a large group of people all over the world. We have a very small core team and then collaborators all over the place. And I want to give you a little bit of information on the story as to how we got here, because I think it'll provide some context as to why we're doing this citizen science project, and why we're confident that all of you going and sampling your homes and working with us to look for these microbes is something that's really promising for fighting climate change, and really actually very important that we do it. So go back, so rewind in time to September of 2021. I was sent this paper by a friend called Volcanic CO2 Seep Geochemistry and Use in Understanding Ocean Acidification. This was describing these shallow marine carbon dioxide seeps in the Aeolian Islands off the coast of Sicily in the Mediterranean. These seeps are basically in, like 10 feet of water, 10 to 40 feet of water, and they are spewing carbon dioxide into the ocean because they're right next to a very large active volcano. Volcanoes produce a lot of gases like CO2. And these seeps, this paper, the Sicilian group, were using these CO2 vents as models for ocean acidification, because ocean acidification is basically when you have Things like carbon dioxide build up in the ocean acidifies. Think about getting closer to lemon juice, right? And that affects the life living there. It affects coral reefs, it affects fish. And the authors were studying how these different gases influence the ecology surrounding the seeps to think about if the ocean were to acidify, what would happen to life in it. However, being a microbiologist, I saw this and I talked to James and Krista, the other co-founder of Two Frontiers, and we thought, well, we know microbes are nature's alchemists. I wonder if anyone has ever looked for microbes that are really good at carbon capture in these seeps. And these are just some details on the vent conditions. Think about it like actually inside the vent itself, which you'll see in a moment. It is truly getting close to lemon juice, basically. We want to know what lived there. And so we wrote this grant. We reached out to the researchers, Marco Malazzo and Paolo Quattrini, great scientists at the University of Palermo. And we said, hey, would you like to go sample these seeps and see if we can find microbes that are really good at carbon capture? And we wrote this grant that admittedly is, it's kind of a crazy sounding title. It was A Tale of Two Frontiers. Naturally evolved carbon dioxide remediation for Earth and space-based applications. So carbon dioxide buildup is a problem. As we know on Earth, it is one of the causes of climate change in our atmosphere. It acidifies the ocean. On space, astronauts exhale carbon dioxide, and the buildup of carbon dioxide in a small space like the space station is bad for human health. Mars's atmosphere is 95% CO2. If we're ever actually going to go live there, it would be helpful to be able to do something with all that CO2 and produce, say, pharmaceuticals or food or sugars or whatever it might be. So we wrote this big, wild grant, which actually got rejected initially, but just in writing it, we became such good friends with Marco and Paula and some other collaborators that we were able to raise enough funds to basically carry out this very small scale, simple experiment. Go to the seeps, use DNA sequencing to measure what microbes are present. We'll have another webinar at some point on DNA sequencing. Krista can lead that. She's one of the world experts on how next generation sequencing works. But basically, we use it as a tool to determine, based on gene sequences, which bacteria are in a certain place. Then grow microbes from the seeps and then evaluate if they're good at carbon capture. Very simple. We put this large team together and we went there. So this was, September of 2022. At this point. So there's James set up in a hotel room. We've gotten very good at lightweight science all around the world, where you can pack everything in a briefcase and then go anywhere and do science wherever you want. That is the island of Vulcano with the volcano in question. Oh yes, James has a bunch of cases behind him because we're using his office as storage right now. So yeah, there we go. It's amazing what you can do nowadays with low-cost scientific equipment. And so on the right-hand side of that picture, you can see the volcano. This is the island of Vulcano. It sort of smells like rotten eggs. That's the sulfur coming out of the volcano. And the seeps, if you look right in the middle, are where that white sort of bleached rock is. And here is great. That's going to work. I hope you can all see this. This is a drone shot of the actual sampling area itself. So you can see we took samples in the sort of white water. That's all bubbles of carbon dioxide and other gases. And then it's a gradient of low pH, think like lemon juice inside the seeps, out to normal seawater, pH closer to 8. All the way out there at the other end. So we sampled along this gradient and did that process of culturing and sequencing and analyzing and figuring out what was living there and was it good at eating carbon in a biotechnologically relevant way. Another video, I think this is, oh, that's the same one. There we go. Okay, another one is, this is actually what it looks like underwater. So we're scuba diving for this, right? So these bubbles, this is actually funny because we were with a photographer and he got lost because the bubbles were so thick. So he's currently trying to look around and figure out where everybody else is underwater. It was all very safe, like you're in 10 feet, but it's just kind of funny. It's quite loud, as you can tell. And the water is warm coming out of the seep because you're, again, near an active volcano. And this is all CO2. So we were wondering, like, think of, again, a square meter is huge for a microbe. So this is so much area for interesting microbial life to be thriving. Back in the United States, we took samples with Max Schubert and George Church's lab at Harvard. We grew up these green, interesting microbial consortia and eventually single microbes. And green is really exciting. When we saw green things growing in our samples, we got really pumped. Green means photosynthesis. Like plants are green, right? Photosynthesis being sunlight and carbon dioxide powering a cell. And so what that basically means is carbon capture is likely occurring in the organisms growing here. And we found this organism called Utex 3222, which we named Cyanobacterium epininum var. Volcano for the island from which it came. And informally, if you've seen any news about this in the popular media or anything like that, we started referring to it as chonkus because the cells are huge. So you can see that scale bar, 10 micron down at the bottom. I said that most microbial cells are 1 micron. Well, these guys are gargantuan. And that turns out to be very important. Chonkus grows very, very quickly in high carbon dioxide, faster than most other cyanobacterial strains. And again, this came from those seeps that we were diving in. This is, and so Utex 3222, and you can see the most interesting thing here, the left-hand side shows its density compared to other comparison strains. So it's much heavier, again, chunky cells. But on the right-hand side, you can see this GIF that is basically showing that it sinks naturally and very quickly. That is unique. As you can see, there's a comparison strain on the left-hand side. It's not sinking over the same 2-hour period. About 20 to 30% of the cost of bioproduction, the cost of capturing carbon in like a big pond. So say you have a fossil fuel plant that's emitting CO2, and you want to have a pond of algae there, like cyanobacteria that are capturing the CO2. 20-30% of actually that process, of the capture process, comes from having to concentrate biomass and then do something with it. Concentrate being squishing it all together. Chonkus does that naturally. It's naturally evolved to do so. And so this result was so exciting from a scientific standpoint because it demonstrated our hypothesis that microbes are nature's alchemists. You can go to a place where there is a lot of a molecule and they will know how to eat it better than anything we've ever seen effectively. That was our initial hypothesis, and this is evidence that that might very well be the case. And so this one organism, which, oh, and here's some sort of back of the envelope math we did showing how it could be used for carbon capture. So you have a big pond of it, and there it is making a biomass, which you can use for different interests like bioplastics and things like that, or engineering it to produce sugars or whatever it is. And on the right-hand side, we sort of show that one tree captures about 22 kilograms of carbon dioxide a year, whereas 13 liters of Utex 3222 on the far right is about equivalent to one tree, and that takes up a lot less space. So it's really great. And so this kind of showed that, wow, looking in these extreme environments might be a really good idea. And it kind of led to the growth of the Two Frontiers project. We've now run 4 expeditions around the world, ranging from terrestrial high CO2 springs in Colorado back to the Aeolian Islands to Shikanojima, which is off of Japan. We have many more planned, looking not just at carbon dioxide, but at methane. And we're putting all of these samples into an open resource for the scientific community, the 2FP Living Database. We're sequencing everything, characterizing what's growing there, identifying how it's capturing carbon, how it can be used to actually make an impact in carbon capture in the real world. It's not meant to be just basic research, and we're sharing everything. We're a nonprofit, right? So the whole plan is to basically make this available to the community so anyone who is studying carbon capture can work with these organisms. But what brought us all here today is this 5th initiative, right? This 5th project in the Carbon Initiative, this citizen science work that you all are very wonderfully involved in. I can't emphasize how excited I am to have you here and to be doing this work with you, because I think we are going to find just as promising organisms in the Extremophile in the Home and in the Wild project as we did in Volcano. And so, and all these other samples as well, all these other sites as well. So let's just take a few minutes and talk about this, and then that's basically all I have. We can open up to questions and conversation, but effectively we're going to talk about exploring the extreme microbiology of your home. So as we established, the Two Frontiers project is about scientific exploration from the depths of the ocean to the reaches of space, and now in your very home you live in. Because individual microbes may be invisible, but they leave very visible marks on the world. So extreme microenvironments in your home may hold organisms that are just as effective at carbon capture as Chonkus, and you would be able to identify those microenvironments by signs of microbial life we are all used to, like strange slime in your sink, or something odd if you haven't cleaned your shower in a few weeks, or something in your air conditioner drip tray, or whatever it is. Those slimes and groups, and those are groups of microbial communities that have grown to the point, they're not hurting you, they're not doing anything, they're just living there. But they're often living in extreme microenvironments where they could be really good at capturing carbon. And because there are enough of them, you can see them. And so the whole premise of this project is let's look around our homes, document and see what potential microbial solutions to climate change are hiding under our very noses. And then from there, actually take those samples and do the same thing that we did with the alien island samples previously. So some inspiration on where to sample. We can talk more about this, and James can jump in whenever you like. But we list a bunch of things on the website, and we're really interested in what you might consider gross. One thing you have to know about microbiologists is like where we get really excited about gross, smelly slimes. And so if you see that around your house, like let us know. And so effectively, we're really interested in HV, like air conditioning, drip trays and drains. James, what's the name for that in the third image there that I hate so much? It's what is it? I can't hear you. Oh no.
Sorry. I apologize, everyone. I was talking with someone who worked in the HVAC industry, and that sort of reddish color that you see in the second from the bottom image there is a drip tray underneath a air conditioner. And so what occurs is water condenses out of the air and drips down, and there's almost no nutrients there. There's no carbon except for the CO2 in the air. And these trays often have iron in them. It's metallic iron or in a reduced form. And so there's organisms that can actually use oxygen, use the energy from oxidizing the iron, just like we use the energy in, the sugar in the Snickers bar, and they fix CO2 out of the air. This is one of those amazing capabilities that Braden was discussing before, and the HVAC folks know about this. They don't know about the microbial details, and they call it pan pudding, which is sort of awesome.
I don't know why. Yeah, so pan pudding. We're really excited about pan pudding. If you see that, let us take a picture, upload it to the site, and then we'll probably send you a sampling kit. Other stuff we're excited about, water systems. So hot water heaters and shower heads that have unusual slimes in them. So James, I'm going to call on you again because you have a story that from a paper published many years ago, I believe, that talks about a showerhead slime that James and I have been friends for over a decade now, right? And he's been talking about this paper for years. And so if any of you have a showerhead that looks like that, please, I know one person does, that came from the program, but send us more. But James, can you please explain why that's exciting?
Yeah, again, this is a super nerdy thing to get excited about, but there's this old paper, and Sarah wrote it up in a newsletter. You can find it in the chat function on our SitSci page. If you read the paper, it's a sort of dry scientific paper. But if you read between the lines, it's a hilarious story because a member of the community brought a showerhead in to the local university. And it was growing like little tendrils out of the showerhead, and they just kept coming back no matter how much this person cleaned it. And so clearly they were horrified by this thing in their shower. They wanted to get rid of it. And the scientists that they gave it to were, of course, fascinated with it and showed that there were some organisms in that slime that were fixing CO2, again, very low nutrients, and turning it into that material, the sort of goo, these little tendrils that were coming out of the showerhead. And that's actually an amazingly useful thing for a microbe to be able to do, capture CO2 and turn it into a polymer. And it was from some time ago, they didn't have some of the tools that Braden talked about that we're able to bring to bear on these, sequencing the DNA in the actual organism, new ways of culturing them. And so that knowledge, scientific knowledge, was lost. And I would love to find out what those microbes that can make slime in nothing but air and clean water, what they're making and how that could be useful.
Wonderful. And then the other thing that we want to talk about is solar panels. Because of exposure to radiation and sun, you basically have high concentrations of sunlight. Remember how I said photosynthesis is something we're really excited about? And then James, would you like to add to that?
Yeah, this is, again, you might not think of solar panels as an environment or an extreme environment, but it is, both because of the light and UV and the heat, but also because in many parts of the country, they're pretty dry. And so it's another example of a place that could hold some really important organisms that have maybe characteristics of being able to withstand desiccation and dryness and fix CO2 even under those conditions. And then the last thing here is this somewhere new. We've talked about some things that are known and from previous research. Oh, I want to call out another one, the hot water systems. We'll be sharing some information about how you can drain the sludge out of the bottom of your hot water tank. And there's hints in the scientific literature that there's some very widespread CO2 fixing organisms that can survive inside a hot water tank. They just sort of attach to the sides and fix CO2, the very small amounts of CO2 that are in the water, and they can grow at the temperatures hot water, a hot water tank, which is sort of amazing. It takes a little bit more effort to sample, and we'll be sharing some information about how you might go about that. This is just what's known. Those kinds of extreme environments and the microbes in your home. What we're really looking for is to hear from you all about interesting things you've observed that seem to be alive or microbial, slimes and goos, all the things that environmental microbiologists get so excited about, and see if there's a place that maybe no one has thought to look before. And you could be involved in the discovery of a new kind of extremophile environment, or maybe even new kinds of organisms that we're growing in your home.
That's right. And this is basically recapping what James said, but just to sort of hammer it home, we're really interested in things like air conditioner drip trays, shower heads, refrigerator and freezer drip lines, washing machines, because of the fluctuation in temperature and the wet and dry cycles, and then dryers, of course, microwaves, solar panels, really any of these little environments. So if this sparks anything, we'd love to hear about it. We can talk about it. In terms of how this works, very simple. First thing is document. We have the websites live. I think nearly everyone here is signed up. You can take a picture of an interesting microbial sludge or environment, upload it, and then we'll keep working with you and thinking, oh, that one's really interesting. That one we think we have already or we've seen before or whatever. And then in the next couple of months, likely in the next month, we're going to be sending out collection kits. And so that'll be based on, we only have a limited number of kits we can send out, right? So we're looking to collect a set number of samples at this initial round that are effectively we'll send out little tubes and give really detailed instructions as to how you can collect your microbe. Again, these are not pathogens, but we don't want you touching them, right? Because we don't want to contaminate with human-associated microbes. And then we'll do some work culturing and DNA sequencing. That's a DNA sequencer, a nanopore device on the right-hand side, and analyzing. And then everything we learn we'll report back to you. We'll include everyone as much as possible in every possible way of the scientific process. If you want to be involved in writing papers, please reach out. If you want to be involved in anything else, please reach out. This is meant to be very much a community building process and something where everybody is involved as much as we possibly can have everybody be. And then, so this is what the SAMP campaign site looks like. So you have 80 people signed up, 26 sets of unique observations by different people. That's honestly amazing. I was hoping we'd get above 50 in the first couple of weeks, and we shot past that. So that is really wonderful. I would love to see if we can break 100. So, I officially deputize everyone on this call. If you have friends or family or anyone who has an interesting house, or if you have that one really gross friend before when I was in college, that was probably me. I wasn't, cleaning the sink enough or whatever. If you have that one really gross friend who has a strange thing growing in their shower or whatever, toilet or whatever it is, please get them to sign up and have them take pictures of their samples, because we're really excited about all that unusual stuff. In terms of, there are a couple of things you'll see it listed on the website. We don't want human associated samples. We don't want anything that came from really a living organism or a person. We were not set up to process that, and where you need a lot of basic, very formal paperwork and forms and insurance and liability stuff to be able to do that. So we're focused exclusively on environmental microbes. And then, of course, and then, I don't know, where am I? Well, okay, great. And yeah, that's I think about it. If you have any questions, we can now chat about it. We can talk about interesting sites to sample. I'm going to take my screen down, and there we go. Yeah, that was almost exactly 30 minutes. Wonderful. And so thank you all so much for coming and listening. This is truly a wonderful turnout for a midday webinar. And I don't know, Sarah, do we have any questions or anything in the chat?
We do have one question, and while we respond to that one, I'm going to pull up some slides too to show just some of the cool stuff you guys have already submitted. And if there's any logistical questions about the website, I probably forgot to introduce myself because I always do that. But I'm Sarah Newman. I'm the representative with SITSI. And Braden, two things that I took away from that. First of all, scientific emoji. I wish I had that lingo. I'm going to definitely borrow that in the future. I love it. And then the other thing is that this is why I love my job, and I love getting to know all of you. This is a very friendly group. I know I've said that in my many communications to all of you, but I got my start in wildlife management and invasive species ecology, but we're working in this participatory science space. I get to learn cool stuff all the time, every day in new parts of science. And I hope you all are as excited as I am, because this is certainly not my expertise area, but it's so cool and fascinating to see where it is going. So David had a question. He wanted to know if Chonkus can Chonkus capture CO2 directly from the air, or is it necessary to bubble gas with high CO2 concentration through the water?
So the way it works, it would be better to bubble gas through the water, but it's in an equilibrium, right? So if you have, the way you grow it is you have a test tube, right? And you have those test tubes, a little bit of liquid in it, you have Chonkus growing in the test tube, and then you have CO2 in what we call the headspace. So you basically inject a lot of carbon dioxide, high concentration in the top. I don't know, my lights just flickered, that's strange. But you have a lot of CO2 in the headspace, and basically as Chonkus consumes CO2 in the water, because the CO2 wants to maintain equilibrium, the CO2 from the air will transit into the water to maintain balance at that barrier, and then it will be able to eat the CO2 in the water as well. So it is optimized to work in really high CO2 conditions, sort of universally. It's not direct air capture. You've probably seen maybe in Iceland, like there's that company that has the massive machines that are sucking CO2 out of the air. It's not doing that. There is a liquid medium involved.
And we have two other great questions. You might have just answered this one, but I'm not sure. While Chonkus captures carbon efficiently relative to other photoautotrophic microbes, isn't there still the argument that man-made DAC technologies are more efficient? If so, I was wondering what advantages microbial carbon capture offered over, I'm probably saying this wrong, but DAC, and what makes it a valuable pursuit? That is from Mark. Thank you, Mark.
Thanks,
Oh, we've
Mark.
got lots of questions coming in. This is great.
No, that's a great question. So you're asking about the comparison between Chonkus and direct air capture, if I understand correctly. Great. So effectively, it comes down to what you're doing with the carbon. So there is the overall answer to that is there's no silver bullet for carbon capture or climate change, right? Like there's not sort of direct air capture is the best or microbial carbon capture is the best. They each offer disadvantages and we're going to need advantages. And we're going to use all of them to be able to actually do this effectively and remediate gases from the atmosphere and from the water around us. Number one, microbial carbon capture is optimized for production, right? So basically you can do bioproduction relatively easily with things like cyanobacteria. The highest titer of sugars ever produced came from a cyanobacteria similar to Chonkus. And so what that basically means is for a carbon to value standpoint, so capturing carbon and making, say, a product or something useful that is, or like a bioplastic or something that locks up carbon in a different way, microbes are really good at that. There are other things microbes are less good at. So we talked about point sources, right? Chonkus is designed to capture, like, let's say you had gases coming out of an emitter, right? And you're piping those into like a pond or to a closed area or whatever it is. Chonkus will work pretty well in that situation, we think, because the high concentrations of CO2. Potentially more efficiently in that area than direct air capture, but we haven't obviously done that study. If you have really low concentrations of ambient CO2, like just in whatever, and wherever they're setting up DAC devices, then Chonkus might not be appropriate. Other microbes might be, or other forms of carbon capture might be. So you really have to tailor the solution to the problem.
Awesome
Great
question. I also wanted to let everyone know I did enable cameras, so if you do want to share your beautiful faces, you are welcome to do so. This is a great time to get to know everyone. Okay, so another great question from Victor. Did I ask this? Okay. Are you guys doing any metagenomic sequencing and trying to assemble MAGs, or only interested in culturing individual strains and sequencing their genomes?
That's a great question. I skipped over that for this talk just to keep it a little higher level, but we sequence everything. So every single sample that we get back, we do next generation metagenomic untargeted sequencing on. So what that effectively means is, for those of you who aren't familiar with that space, metagenomic sequencing refers to taking a snapshot of all the DNA present in an environment. So imagine that milliliter of soil with that 100 million microbes in it. They all have DNA. Every microbe has an individual genome or a couple interesting DNA elements in it. You then fragment up those microbes, and you use a sequencing device to read the DNA and the sequences that are there. What Victor is referring to is then the downstream analysis after you read that. So imagine like you get a Microsoft Word document back that has literally tens or even hundreds of millions of lines that are just ATCG, 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 little fragments of DNA that you have read. That is what I get as a bioinformatician when someone who's done DNA sequencing, like Krista, hands me that data. De novo assembly and then binning a MAG or a metagenome assembled genome is taking these little fragments of DNA that we get in our Microsoft Word document and trying to piece them back together like the most complicated puzzle in the world. Trying to say, oh, look, I have 150 base pair fragments, so 150 A's, T's, C's, and G's, the letters that make up DNA. And then I have another one over here that overlaps with it slightly. I'm going to piece it together, and I'm going to do that millions and millions and millions of times over. And that is something is honestly more my specialty than anything else. And that is something we do on every single sample. So every single sample we come back, we have custom assembly pipelines. If you're familiar with like MegaHit, we have a modified version of that works a little bit better for pulling out mags. We then do co-assembly and do all this crazy stuff downstream. And that is important because once I have a microbe's genome, I can look in it and say, oh, look, this microbe has a gene for sequestering carbon. I think this thing is photosynthetic. And then I can go back to the sample that we have banked and I can say, I think I can grow this microorganism that I've seen in the bioinformatic data based on the genes it has. So that's a great question.
They just keep coming. There's good ones here everywhere. So Eric had one. Do you think Chonkus evolved in and around that CO2 seep or evolved somewhere else and found its way there?
Good question. That's such a good question. Thank you, Eric. James, do you want to take this one?
Yeah, absolutely. So we don't know yet. But part of the reason that we're looking at these volcanic seeps that are spread all over the world is because if we can find hints of this Chonkus-like adaptations in some of these other locations, and the DNA changes are the same, we can infer that there is a global spread and that these organisms had some point of origin separate from these sites. If, however, we find no sign of the organisms like Chonkus in any of these other places, and only different kinds of organisms, different cyanobacteria, may be filling that niche, or different kinds of mutations, adapting them to that environment, then I'll tell you that it's actually separate evolutionary paths in each and every one of these CO2 seeps. We don't know yet, but we're collecting the data so that we can find out.
And James, I think this might be a question for you as well, but Braden, obviously pop in if you want to. Mark asked a great question. How do you ensure that unsupervised citizen scientists provide uncontaminated samples that are credible enough for publication? It's always a good question.
That's great. We take a couple of approaches here, and motivated by the idea that to get the kind of reach and look in all of these unusual places, we just simply can't do it without help from a community, is really something, and just letting people participate in this kind of science is really important to us. Very technically, how we avoid this is we do provide information and like gloves for people to wear at least, and some guidance about how to collect things carefully, and Sarah helps us communicate that. And events like this, speaking to the community, you guys will know that this is important. And when we sequence it, there's been a lot of studies of human microbiomes, and even of microbiomes of the built environment. And so it's actually using modern bioinformatic techniques. It's doable, it's achievable to identify when something is coming from an environment that's very well described. like human skin, and you can just throw that data out. We get so many fragments of DNA from these kind of technologies that even if you have to throw out a significant fraction, set it aside, potentially coming from the person sampling it, that's a perfectly reasonable thing to do with the data.
The other thing I would throw in there is the sites that we're really interested in sampling are higher biomass sites, such that you saw that pan pudding, right? It's like orange slime. The number of microbial cells on your hand is going to be much, much lower than the number of microbial cells in that orange slime. James can always do this back of the envelope fast math a lot faster than I can, but we'll just say much, much lower for now. To the point where bioinformatically, I can very easily, as he said, or relatively easily pull out the human contaminants. There should be a small fraction.
That's awesome. And Tom had a great question also about air purifiers, and was wondering if anyone had sent in samples from filters of home air purifiers. Would we be interested in them? James, I'll let you answer that one.
Yeah, absolutely. I think we want to hear about it. We want, if you see something that's unusual in those kind of environments, make an observation. That's really helpful. That's how we're going to, keep our eyes open and find things that we didn't previously know about. And for some kinds of air filters, I expect there to be lower bacterial load. Maybe I'm wrong. Show me something, growing and slimy on an air filter, I'd be very interested in hearing about it. Because a lot of life needs water, at least intermittently. And the organisms that grow in the desert, even on grains of sand, they're sort of existing in a single monolayer of water molecules that are sorbed to the surface of grains. So when you look around your house, I guess my first instinct would be to go to places where there's water. If you think about it, sort of the ecosystem of our house, where water and energy is cycled is often through things like, our HVAC systems. So I think you're on to something by looking within that system, but someplace that maybe had a little more moisture would be even make a even cooler observation. Does that answer your question?
Yeah, that's a great answer, James. And I would also recommend, one of the beauties of SitSci is that we're set up to take images and data, and it's all relatively low cost. And so when in doubt, take a picture of it, upload it, put in some notes, and we can always not sample it. But if we never see it, we'll never know. So if you're not sure, just do it. And there's no harm in that, and only possibility and only potential. So I think just go for it, and then we'll see if it looks cool. James will tell me for sure, or one of his students who's going to hopefully be helping with us with the project. So I think that covers all the questions. Oh no, just kidding. Another one came through while we were chatting. Excellent. Again, from Tom, will there be initiatives also to search for microbes which consume methane in the future? Oh, good question.
Yes. Yeah, we are currently in the process of getting our sequencing data back for all of our carbon initiative expeditions. And so the sort of 2FP timeline is over the next 6 months or so, we're going to be analyzing and really getting deep into that data, looking for new mechanisms of carbon capture, understanding, characterizing more isolates. So, that Chonkus paper was the first one, right? And then we're going to have many, many more, I think. Actually, we already have a bunch sort of lined up. And then as we start planning and moving into expeditions in the coming year, 2025 and 6, we're going to be looking more into other greenhouse gases. One of them is methane.
And another great question from Mark. I saw a new agricultural tab on 2FP's website and was wondering if we could kindly get some information about this new pursuit and its current status. And then we have one more question, I think.
Yes. So that is our crops initiative. We have our carbon, our coral, and our crops initiative. I don't know why, we just sort of are stuck on the letter C. We're gonna have to have a clinical one at some point, but I guess consistency is good. And so our crops initiative, actually, we have an expedition to do some sampling coming up relatively soon. And that is focused, it is funded mostly by a grant from NASA that is trying to grow crops in lunar regolith. So NASA wants to grow things on the moon. And we basically said, let's go to a place on Earth that has sediment and or geochemistry, kind of like the moon, or as close as you can get in some dimensions. And we're going to go and find plants that are growing there, and then we're going to find microbes that help those plants growing there, and we're going to put those microbes on model organisms and eventually crops while they are growing in simulated and then real lunar regolith, and see if they help tolerate it. And that's great because space exploration is sort of this, I think, unifying, exciting thing. However, it is also relevant to helping crops be more tolerant to increasingly desertified environments on Earth. So as the climate changes and we have less rain, we need to find ways to stimulate crop stress tolerance to those stresses. So effectively, we're really excited about kind of the dual-pronged approach here. We have moon, space, exciting, and we also have practical helping advance agriculture on Earth. Again, using this principle of looking in extreme or unique environments.
This is so great. So I'm going to pause the questions there and just quick show something also related to a question Alex had about his sample from or his observation from Germany. So let me see if I can just, too many screens, not enough hands. Okay. Share, here we go. Okay. So I'm going to skip over what SITSI is because I think a lot of you have heard this or have figured it out by now. But essentially, we are the platform that's helping support the data, and this is our mission. This is this kind of stuff we love doing. We're all scientists that got into this because we had lots of questions about cool things that folks like you could help with. Just pointing out that we do have a mobile app. It's not as full featured as the website, but if you wanted to use it, it is out there and free to download. And then Alex asked about his observation he sent in from Germany. I believe this is the one right here. So it does show up on my map. If you're not seeing it, maybe you could send me a screenshot. I'd love to see it. But already, you've all sent in observations across the continent of the US, Hawaii, and now Europe as well, which is so exciting. It'll be amazing to see how this grows and how many more things come through. Just super fast, I want to remind you that you can explore this data, and I encourage you to do that. You can click on different observations and see what people are submitting. You can look at all the resources. There's been a few really cool articles already. We're trying to make more resources for folks to help with the sampling process and kind of the brainstorming about where to look in your home. And those will be here as well. So I encourage you to check that out. And then this forum piece, I cannot emphasize enough how many amazing scientific discoveries come out of groups like this talking to each other. So please don't let me be the only voice on here because I don't like listening to myself talk, but I definitely love hearing and seeing these conversations like what you're doing right now. This is how so many amazing things have happened. Other citizen science projects, there have been cool publications from folks that made a quirky observation or asked a question that this looks weird to me. Like, what do you think this is? And publications have come out of that. So please definitely join into the conversation and ask questions. But this is what I really wanted to show. So already on the results tab, we're seeing in real time the answers to some of the questions you all are answering. But out of the samples we've gotten so far, I thought this was really cool. We've gotten quite a few out of dishwashers, a few shower heads, washing machines, refrigerators, and then this other 50% already. So that's kind of fun. And I thought this texture one is really cool because I was learning a lot of lingo with James when we were figuring out how to ask this question, stringy, slimy, crusty, biofilm. So very interesting distribution of observations here and what people think they are. Yeah, go ahead, James.
this is what Braden mentioned about how through just visual observation you can learn something. The colors and these descriptions, we can look at some of these characteristics and say, oh, that's something that is known before. That's something that isn't. There's sort of, these are hopefully useful keywords that citizen scientists can help describe something in a little bit more systematic way, but it allows us to kind of line it up with what's known and what's unknown, and what would be worth sequencing and might be something really novel.
That's great. So that is all I had on that piece. And I think we are going to wrap it here because we are just a minute before the hour. That's like the most well-timed webinar I've done in a really long time, I got to say. So thank you all again for coming. As Braden mentioned, we might be doing another one of these. I'd love to hear from Krista on all of her amazing skills. And lots of cool stuff yet to come. So please continue to be a part of it. And encourage your friends. And if you work at a university, encourage all those students with gross living conditions to share what they find in their apartments, right? So
yeah. And let's continue the conversation. Let's continue the conversation on the forum. If you had a question that didn't get answered, or you want to ask some follow-ups after you've looked at some of these samples, or thinking about some of the kinds of samples that we're looking for, please just continue on the forum and we can talk that way.
Yeah. Braden, any last words?
Well, thank you all so much for the questions and enthusiasm. And I hope to talk to each of you individually soon. Really, it's really fun to get to share this. So please do not hesitate to reach out and grow the Citizen Science Network.
Excellent. Thank you all. Have a wonderful day.
Thanks, guys.
Thank you.
Bye-bye.